although technically we start at 5 o'clock, but Fellman standard is 5:05.
Sure.
So I will probably give you a very long introduction that gives more people time to come.
Okay.
But I expect we'll have a pretty good turnout.
Okay. Neat. Thanks, kiddo, and thanks for organizing everything. Jan was so fun to talk to.
Yeah, she's awesome. She's my baby.
James and Fiona.
Really? Still want to get this.
Okay. Picture. That's brilliant. Wow. Okay.
I'll give you a heads up.
Okay.
He's gonna sit in the front row.
No. I'm just want to come and say hi.
Hi.
How are you doing?
I have you as Michael up there, but everybody knows that. Good to see you.
Good to see you.
So I'll probably do my introduction from down here.
Okay.
Simply because I'm too lazy to go all the way up.
It's more closer to us.
Yeah, and I'm just a teaching here, so I have to do this room too, so just need to have a bit of fun.
Okay.
Is that okay with you?
We do.
Extra minute. More money. Let's see if anyone forgot any pens up here. There's one. And another. I usually do forget that.
Okay. Are we gonna do the mic thing? Hello, hello. Sorry, Jamal. How can I spell it?
It's 5. We'll give it a few more minutes.
Okay.
Yeah, she said she all class don't work. She didn't even bring like somebody was yes. No, not yet. 3 more minutes. Exactly, your mind.
2 more minutes.
Okay.
I can hardly see you behind this.
I know, I was going to say, I needed like a little stool, but then I'd probably fall off it.
And my offer still stands. I can advance it at your notice if you need me to stand here.
Okay, thanks. I'm kind of thinking I might check my notes now and again, so.
Did you have water? Did you need water?
I don't think so.
Just have to ask you 1 minute.
Thanks. I've got a bottle in there. Maybe I'll just go get it.
You ready? Doctor, you ready?
Yes, ma'am.
Welcome, everyone. It is my pleasure to introduce my friend and collaborator, Lynn Fellman, the one and only. I met her 4 years ago at the National Evolutionary Synthesis Center when I was in sabbatical from Spelman. We were both visiting scholars who had gone there with a particular project. But there, we were both roped into a new project. It was really fun, really fascinating. It was about combining genetics and genealogy in the undergraduate curriculum. Rings a bell, right? I was sought out for my expertise in evolution and education, and she was sought out for her expertise in evolutionary art. So today's event is a result of that association. It is a real joy to welcome everyone, particularly students who have experienced or are experiencing the genetics and genealogy curriculum. So raise your hand if you fall in that category. So this is really designed as an event for you to supplement the instruction that you're getting in the labs. So we're going to listen to a talk titled The Art and Science of You by Lynn Fellman. An artist and a writer, Lynn has a degree in studio arts from University of Minnesota and began her career just like many of us with a regular job and a regular paycheck, she told me. But all of that changed pretty dramatically pretty soon when she got tired of having a regular job and decided to strike out on her own as an independent artist, an illustrator, and a designer specializing in translating complex concepts. She told me she was inspired by her desire to associate with interesting people doing challenging projects. And the topic could be anything. So that's Lynn Fellman for you. The intrepid Lynn was an early adopter of digital tools as far back as the 1980s, creating interactive multimedia presentations for corporations and publishers. She believes that mixing traditional and digital media allows a full expression of complex ideas and is an approach that she continues in her current work. What is her current work? Her current work focuses on creating art designed to understand basic science concepts to guide smart strategies for health, economy, and our environment. She sees that the problem is getting the fundamentals across in a clear and engaging way. And her solution is to combine accurate information with poetic visualization, as you can see up here, combining art and story to illuminate the beauty and benefit of, hold your breath, genomics and evolutionary science. So who would have thought that would come in the same sentence? You might wonder, how on earth did she go from making art for corporations and publishers to creating art for genomic and evolutionary science? It turns out, like many of us, Lynn has had a lifelong fascination for the story of human evolution, the story told by fossils like the hominid Lucy and fossil hunters like the Leakeys, the story that has been told by stones and bones, as many anthropologists and archaeologists say. As we know, that story had a dramatic development at the turn of this century with the completion of the Human Genome Project. That revolutionized science and our understanding of human origins and health. When genes entered the story for human evolution was when Lynn decided to make yet another dramatic career change. She was captivated by one thing, the story of human evolution that was to be unraveled by genes. She was thrilled that the story of human evolution could now be told by bones and stones and genes. She asked, what are the big ideas? What are the ideas that matter? Since that time, which was 2005, Lynn has spent all her time and energy figuring out how art can tell the story of human diversity, human evolution, and genomics. As you can imagine, this is not an ordinary calling, and Lynn had to make an extraordinary effort to answer this extraordinary calling. First and foremost, she had to understand the science behind the discoveries. For any other person with no training in science, that might be daunting, but not to Lynn. She just decided that was no problem. She read a lot, and we tell you to read a lot. And she basically taught herself a whole lot of science by reading books, even scientific journals, and believe it or not, she would go to conferences to read the posters at the science conferences. So she absorbed all she could about genomics through self-study and a couple of science mentors along the way. Next, she had to figure out how her art could tell the scientific story in an accurate yet captivating way. This part, I imagine, was the easy part for her. She writes that as she learned about the field of molecular anthropology, she started to draw, write, and talk about the amazing discoveries in human evolution. The outcome of her research were portraits based on mitochondrial DNA and Y chromosome data from real people, willing friends and colleagues. Finally, and this is the hardest, she had to find her audience. Keep in mind, this was way before STEAM. The integration of art and science, technology, engineering, and math was a buzzword in media and university campuses. I imagine this was the hardest part because she had to make a path where none existed before. She shared stories of knocking on doors and writing to people who did not care to listen to her vision, and how art could enrich people's understanding of themselves, human diversity, and evolutionary story. One story that I will share today is that of her sitting patiently and cheerfully in the office of a community leader who had ignored her emails and phone calls repeatedly. So Lynn just showed up there and waited till this person gave her some time, and at that point Lynn just gave her the book of portraits that she had created of these people with their mitochondrial and Y chromosome data, and that leader was then convinced of the beauty of this approach and the richness of this approach. Her art continues to convey complex ideas in a poetic way, combining engaging stories, vibrant illustrations, short animation, interactive features to educate and delight, as she says. Her work has been supported by foremost science agencies like National Science Foundation, AAAS, the American Association for Advancement of Sciences, by many private funders such as Robert Wood Johnson and Fulbright. She has continued to work with the smartest minds, because that was her first goal, such as pioneers like herself, Nina Jablonski, the renowned anthropologist, Ben Mishmar, the archaeologist who changed paths to become a geneticist, P.Z. Myers, and Eugenie Scott, two very famous science advocates and writers. She has hosted radio shows on science, and she has been featured in radio shows like NPR Science Friday herself. So folks, let's welcome Lynn Fellman to Spellman, and let's listen to her fascinating story.
Thank you, Dr. Pai. Wonderful introduction. I really appreciate it. So, thousands of genes, lots of genes, code for height. You can see where I was, the short genes. But I'm going to stand over here. I don't know if you're able to see me, but you can see the screen. And the mic, are we good with that? Do we care about that, or? Not to worry? We're good. Okay. All right. So, thank you. Thank you for coming on a Friday night, a beautiful Friday evening, to come and hear my talk about the art and science of you. I'm Lynn Fellman, and I'm from Washington, D.C., and I'm haplogroup H. So like Aditi said, I'm an independent artist, writer, and sometimes I do animations when it seems to be the right project, a right fit for the project. And she already gave my credentials, but you can see a few things there. Cold Spring Harbor lets me come and show my work, and I usually come to Biology of the Genome Scientific Conference in May. My Fulbright was in Israel, and that was working with an evolutionary geneticist. But I'm really excited about being a member of the Genome Writers Guild. That's a scientific organization. Ask me about that later. I can tell you something about that if you're curious. And the other group that I'm happy to be a part of is the National Association of Science Writers. I'm aiming for a 30-minute talk. I'll watch for your reactions. If I see a little sleepy eyes, I'll speed it up. But please do interrupt me if you have questions. Wave your arms. That would be great if you got questions. We'll take them at the end too. And on most of the slides, right at the bottom in little type, I have links to interesting websites and other information. So if you want that information, there's my email address. Send me an email, and I'll get that to you. So, how many of you have got your DNA data? Like maybe for genetic ancestry with one of these kits? All right, yeah, raise your hand, okay. To like about, I don't know, half. At least half. Congratulations. You're now participants in the genomic revolution. You are the avant-garde. I got my genome sequenced, well, it was a mitochondrial DNA for genetic ancestry 12 years ago, 2005, and it was with the Genographic Project. That's the black kit way on the end. And when I got that, I was so stunned by, well, there wasn't any surprises at all, really, but just that experience of the swabbing and the getting it back, and then looking at their narrative that they talked about to explain essentially what was deep ancestry coming out of Africa, how we populated the world. I just said, okay, I'm doing something with this. I don't know what it's going to be, but I started to draw and draw and draw, and what I came up with is what I'll be showing some of it to you today, a whole new body of work, just a portion of it, because I don't want to overwhelm you, but then also, specifically today, how that work relates to what's going on here with Dr. Pai and you at Spellman in the biology course. So now, as genome explorers, you probably already know, someday soon, everyone will get their genome sequenced. And at some point, when it's all sequenced, we have the data, it'll really become part of standard medical care. So all of us will be genome explorers. And I use that phrase, all of us, because that's the name of the program that NIH, maybe you've heard about it, National Institute of Health, has to sequence 1 million, probably more, US citizens, you and me, and pair that up with our phenotypic data to understand, better ways to live healthy, insights into risk for disease. So watch for that as that develops. Oh, and why the baby face? Okay, this is a really cool project. Out of Harvard, Robert Green, he's the PI, and his project is called BabySeq. So it's a small cohort of parents who are expecting babies, and then they sequence that newborn infant. So the idea is to understand that early genome and then do a little not a little, but a longitudinal study to see how that changes over time, looking for risk factors for disease. So check that out too. So we all have this unique genome story. And have you ever attempted to explain the science and felt frustrated? It's the science in you, but how do you talk about it? Have you ever tried to explain to a friend or a family member what's going on with that and they just didn't get it? Have you ever wondered how to make your DNA results easier to show, share, explain, understand? All right then, art and story for science. So I found with some of that work that I was developing as I began to understand the human genome and the sequencing data was that when you put a story with pictures, it has a real power to communicate the value and the beauty of scientific research. And what I also found, that when you can show it and share it, your genome story can inspire others. You can make people smarter. You can really get them to be curious. We need advocates for science. How important is this? Super important. Yay, Atlanta! So, is anybody up in that picture? This was from your website, a March for Science. Yeah, did you go?
Yes.
Some of you? Yay, okay, we did in DC too. So, science is under attack. Science and scientific research, you know that. It's really been an issue lately. The EPA administration, current administration, didn't allow some scientists to give their current research on climate change at one of the conferences. So some of those things are starting to happen, and I'm sure you're aware of it. But what we do know is that when you understand basic concepts, all of our citizens understand basic concepts, they make smarter decisions as voters and as citizens. This is something that's really valuable to our society. So now, getting to what Dr. Pai was talking about for a little inspiration, I'm going to show you the DNA Portrait Project. And it was a cohort of people, and I'll give examples from that and tell the story behind it. And then towards the end, I'll explain a little bit about the project here at Fellman, and then how the two are interrelated. So, some background on the project. It took place in Minneapolis, Minnesota. The University of Minnesota had a brand new building on the north side of Minneapolis, kind of known as a tough neighborhood. But the thing about the neighborhood, it was really ethnically diverse, really interesting, very vibrant. But this building was put there by the university for research and resources for the whole neighborhood to come and work together, and classes and that kind of thing. So the building was still underway and being designed when the director, Dr. Irma McLaurin, had seen some of my work. She and I got together. She's an anthropologist. And she said, let's find a way to celebrate this diversity in this community. Let's do something with this DNA portrait idea where we get people in the community, have their portraits, and then showcase it in this new building. Okay, so I'm jumping ahead here a bit, but when this exhibit was put up, I was really stunned. I was stunned by the reaction because I hadn't really experienced this before. I stood back along the walls and I just saw how people looked at the pictures with the stories. It really changed my life because I saw the power that words and pictures together, and it wasn't just the art by itself or just the story by itself, but it's the two together, and most importantly, it was really personal information, family stories, genealogy, with that deep history, that deep ancestry story that was just starting to get known. The year was 2010 in our culture. The people. Okay, this is a big group. It looks like a big group, but the group in there is some of the people who are supporting for the exhibit helped to make that happen. But you'll see the director there, and then 5 of those people were the people who I call my portrait participants. And here they are. I'll show them to you one at a time and say a little bit about them. So again, keeping in the idea of the demographics of this community in North Minneapolis, to make sure we got a good balance and a good representation. So first of all, let me explain how it happened, because this was the key part. Is Dr. McClure said to me, Lynn, we need to first identify the leaders in the community, leaders of organizations, and go to them, talk to them, explain what we're doing, which is what I did, like Dr. Pai was saying, show my book, and then they would nominate or invite somebody from their community to have their portrait done by me. Okay, so that was the premise. That's how we started out with the process. And that's how I got to meet these people. So Kao is an American citizen, U.S. citizen, originally from Laos. Riva is Jewish. Up in North Minneapolis, there was a sizable Jewish population. Wayne is African American, and as a community leader, he was in charge, or had started a lot of men's group. A lot of it was about anger management. Really interesting fellow. So he dealt with a lot of people who came out of prison. A good-hearted guy. Judy, African American. She was leader of the Head Start program in the community. And Ron Bakanaga, and that's his mom, Gertrude. And so Native American from the Ojibwe tribe. So what started out with this wonderful group of people, I call them my brave genome explorers, because they didn't know much about science, specifically genetics. They were taking a leap, and they were really trusting me and really willing to be part of this. But they were excited. And what I found too, that being part of this, they learned about themselves and the science and really became advocates slowly but surely. So, to really get things started, I bought the kits, had a pile of them, and then after I had met the leaders and they nominated these 5 people that you just met, I invited each of them to come to my studio individually. And they sat in my red chair. And we talked, I took notes. I had to get to know them to really know who they were professionally, like the leadership positions. What did they do? Tell me more. Their family stories. So this would be the genealogy piece, the current ancestry story. And then we opened up the kit. I helped them take their sample, and then I mailed it for them, make sure it got into process. And I took some photos to get my drawing started. As you know, these kits take a while, so I could be drawing the portraits while these things were going to the lab. Science in the project. The genographic project, so this is all about changes to populations over time, human evolution, the anthropological approach to this, where they map the haplogroups across time and across geography. The PI is Spencer Wells, and it was started in 2005, April of 2005, and I got my kit September 2005. That's what those kits look like. They're kind of rudimentary looking. And this was the map that they put out. So this is a historical document. We might call this old data, but essentially that information has stayed the same. And we all know that it's blossomed, it's grown, it's been added to. But that is pretty much fundamental good knowledge that has been built on. You can see the yellow and the blue lines. So the yellow lines are the female story out of Africa, mitochondrial DNA, and the blue is the Y chromosome, male and female. Okay, so this is something I've wanted to do for a while. I don't know if I got it quite right, you can tell me, but I really wanted to explain the founder populations. So on the far left, there's the big group, and I'm thinking maybe still in Africa. A larger group of anatomical modern humans. A small group breaks off, hang out together for a while, mix it up a little bit, new mutation occurs. Another group breaks off. They keep the blue, but they add the red after a little while. And as you can see, I want to say a smaller group, and they're not showing less genetic diversity, but this simple sort of approach. And then the two other groups, you can see how the mutations are carried out. What I really wanted to do was animation to show how this also shows frequency in the population. Am I missing it? Do you think I got it right? Pretty much? Okay, tell me later. All right, something else, a little bit more of the science. I know a lot of you really know this stuff, but this was the kinds of things I got to tell the 5 people that I worked with. I got to tell them some really basic science, and I did it in little small snippets as they could take it in. Well, you know, we're all like that. But one of the big ideas that was hard for me to get right away is, this is our genome. Our genome is made up of kind of 2 big pieces of DNA. We've got the mitochondrial, or an organelle, in the cytoplasm of the cell. That's the female lineage, passed mother to daughter. And then we've got the other 23 pairs chromosomes and the nucleus of the DNA. DNA in the nucleus. Chromosome, you know where they are. All right. That's what I tried to explain to them. Because they got the kit, and this very early kit, you had to check off the box, male or female. Why is that? In fact, why were some just getting one piece of information, but the others were getting both? That was part of the story too. Y chromosome, mitochondrial DNA for a couple of the guys. So that's a karyotype. I don't even know if we're looking at karyotypes anymore, but you can tell what gender this is, right? The curious why. So short time to repeats are a different kind of mutation, but that's what they look at in the Y chromosome to get the information, compared to the mitochondrial, which is a SNP, or a point mutation. So this is different information. There's a little baby Y chromosome over there. Yeah, and the reason why I say it's curious is because the DNA is coded in these palindromic repeats. So palindrome, you know, in literature, it's read front to back the same, and that's what happens with the palindromic information that's encoded. And in fact, they've had a lot of trouble early sequencing to understand the genes in the Y chromosome because they didn't know where the genes started and ended. So I just think that's kind of poetic. Okay, so I'll talk about just 2 of the people and the portraits that I did for them. Judy, like I said, director of Head Start Group. And then later on I'll talk about Ron and Gertrude. But Judy, here she is, close up with her portrait. So you can see the storyboard behind her and the portrait then next to her. And if I got it right, this was like one of my tests to see if you can tell who she is. It was an artistic challenge. I didn't know if I was up to it, so. And I thought you'd like to see just a close-up. So this is a combination of digital, using the pen tool, with brushstrokes, pen and ink brushstrokes on paper, moving large to get that energy in the strokes, and then bringing them into the computer, and it all ends up as a digital image. But you can see how I've got layers of translucency going, and when you get that big on the screen, those eyes, it's just mesmerizing. It's so cool to work on it. All right, so she is. I titled this Spiral Journey. Yeah, Data was Genographic Project. Her haplogroup is L3 with a subclade. So all of us in this room are L3 probably? Well, we're all related, but most of us are L3. I'm L3, and then my group, way back L3, of course, headed out, went north, because I'm Western and Northern European, haplogroup H. But as an African-American woman, there was no surprises here. This is what she knew from her family stories as well. But when we talked about it, I mean, it's a wondrous story. There was a population that stayed in Africa that eventually some of them, over time, became part of the African slave trade. We know that story. And a small portion then, of course, headed north and went out of Africa. And as they picked up those mutations eventually, yeah, I'm way up there somewhere. But we all share L3. In fact, today at lunch, who had the L2? You're like ancient. Really ancient. It's so cool to see that because your little haplogroup is like boom, boom. That's all you are. It's really cool. When you look at the Genographic Project, they give dates to these haplogroup mutations that are progressively spaced over time. 67,000 years ago, L3.
Lynn,
Yeah.
can you explain some of the other details on this image, like the maps and the spiral itself?
Well, I added the spiral because so many of us stayed in Africa. A lot of us stayed in Africa and mixed it up. So admixture, you've talked about admixture in class, right? And we are just beginning now to really understand the extent of that. In current populations, but also in these ancient populations where the morphology of the different hominid groups, modern hands and feet, but a really small brain, all kinds of this sort of mixture, and that's why I had that sort of circular kind of thing going on. And I've got some chromosomes in there, and the helix dances across the top. I work a lot with colors. I like the idea of mixing water with a landscape. Some of the other things I was thinking about was how to show who she is and what she looks like, but kind of push her into the landscape so she's actually part of it too. I kind of didn't know if there was a sweet spot there, but when I printed these, I did all my own printing on a large format printer with a really yummy rag paper and these gorgeous pigment inks. So these prints had real different feel to them originally. You could see, I think, a little bit more of the layers. I have no intentions for you to try to read all that little bitty text, but what I wanted to show is the three main pieces of information I was talking to on all of these storyboards. It was their professional association in the organization in the community, and these people had stature, and they were well known. And then their family story, they were just really generous about that. And of course, then I explained the genetics, what's happening with that haplogroup. And up there, that circular shape, so you know that's how mitochondrial is shown in that graphic symbol. And it is because it is sort of a circular chromosome is how they consider it. Just another close-up to that. Okay, so a story about Ron and Gertrude. Dr. Pye talked about it a little bit, but this was really interesting because I was aware, even then, and well, all of us were, that Native Americans were really skeptical about getting their DNA. Well, they had been abused. It wasn't used wisely. They weren't informed. But there is a sizable Ojibwe population, Native American, in the North Minneapolis community. I really wanted them to be part of this. So I had the name of Gertrude Beckanaga as the leader of the Upper Midwest American Indian Center. So I was to contact her. Well, I sent her an email invitation. I left phone messages. She didn't answer either of them. What should I do? So Dr. McLaurin she suggested to me, go to her office, hang out, bring your book. So I did, and I thought, what the heck, chill. It'll work out or it won't. So I sat for about 45 minutes. Finally, she agreed to see me. She had a really nice office, big desk. She was looking at a lot of papers. She kind of told me to sit down, and I just waited for her attention. And then I opened my book. I had a book I had printed with other examples, and I laid it in front of her. And I just was quiet, and she looked. And then she looks up with me, and her eyes are bright. She totally got it right away. She was really excited. The reason why she was excited is she was into genealogy. She had the French explorers that came through the St. Lawrence Seaway, I want to say freeway, and she knew about those people. She had been following some of the genetics. She was really excited. She says, oh, This is wonderful. My son's got to be part of it. She picked up the phone. She called up Ron, who was down the street. He had built up this charter school for, it was middle school kids, Native Americans. Anyway, so what we decided to do is I was going to do Ron's portrait, but I got a kit for both of them because Gertrude then could have that experience of doing the kit thing and then be part of all this. Okay, so that was that fun backstory. There they are in my red chair. Here he is standing next to his portrait. Really good guy. I had fun with this. Oh man, so I thought I'd step this through. So I work in layers. Each of these portraits maybe had 12 layers. This is Adobe Illustrator. So as you as scientists, I think you use Illustrator quite a bit to do your figures, your charts. Yeah, it's tricky. But I love vector art because it's resolution independent. You can scale it up, scale it down. You won't lose resolution. Anyway, so I'm starting to show you some of the layers. And I really like the idea of translucencies, wanting to give you a sense that it's not digital art. Maybe it's watercolor, maybe you don't really know what it is. Okay, so that's sometimes what I'm working on in that preview stage, and then I'll go back to the color back and forth. The title of this is Crossing Beringia. I call it an archival pigment print. So this really is archival. These pigment on these papers will last 100 years or more. And what I'm showing then is really, this is a brief, sort of simplified version of those sweeping groups that came over that lost continent of Beringia. So I'm not 100% accurate, but it gives you that sense right there of kind of that amazing journey that these people made. All the way down to the very tip of South America. And they are still thinking that window was 16 to 18,000 years ago when they made that leap. So when the exhibit went up, I saw something that really, like I said already, pretty much stunned me. I stood in the back of the wall and just watched. And this was a brand new building, so there was more going on than just the exhibit. So a lot of people from the community and the university, so the provost and all these folks came. But what was really fun, as I sort of stood back and watched and listened, is that the 5 participants, the portrait participants, became celebrities. Because people who didn't know them, they'd say, "That's you up there." So that was a really fun thing. And then something else that I saw that happened was that Ron, he was really the one. He stood in front of his portrait, and he had students there. Students were in front of him, and he went on to describe what was happening in that travel from Asia, coming across Beringia, and he paired it up with a little bit of the DNA story. Details weren't, but then with the Ojibwe creation story. So he melded the two and just kind of gave his own rendition of it. And it was just wonderful. Something else I saw, people actually read this stuff. They stood there and they read it, and they got in these small groups, people who didn't know each other, and they start talking about this. Later, I was told that science teachers would bring their classes. It was up for 6 months. So, like I said, through this work, I really became a science communicator, understanding that there is power here with these stories we can tell about ourselves, share with others. Okay, a short bit about this wonderful genetics and genealogy project here at Fellman. It's right now, it's a component that fits into an undergraduate biology course. But it's really to talk about some key ideas about human evolution. And I know we can talk more about that later, but what I was asked to do is to create sort of a visualization tool that would allow the students to incorporate the DNA data they was getting from 23andMe, so their own information, somehow to bring the pieces of information of that into their family tree stuff and create something new to tell that story, to explain it. And of course, you know what we're trying to do too is to like reinforce these learning objectives. When you work with different materials and you've got your kind of face close to it, it's a different kind of way to learn about this. You can kind of integrate it. All right, so that was the idea. And what we found from the second year of modifying what I originally started out with was a PowerPoint application. And it had a bunch of different components or modules. But essentially it was some instructions to give a few pointers of how to use this. Also like where to find information out on the web, how to do it carefully so you give credit. That was very important. I gave a couple of samples. So who wants to start with a blank canvas? It's really tough to do. So some other student examples. A ton of graphic images. Some of them I had drawn, but others are royalty free. So we checked all that out. Some basic trees you could build on. And how could you not have skulls? I mean, look at these. These are magnificent. They're from the Smithsonian, and they freely share these. So Cro-Magnon and Neanderthal, those are there. Okay. So now that I've given you some ideas, I know you all are thinking about the students here that I've already met. A lot of you at lunchtime, you've already figured out what you can make. So, but the question is, what could you make? So, whatever you decide to do, or however you tell your story, just do it in your style. I've got my thing, you've seen my thing, but there's lots of ways. A collage, a poem, a composition, you get it. Just make it your way, with your voice. Because it's really important to be an advocate for science. When you tell your genome story, your personal story, and connect it with the science, you can inspire others, you can make them curious, you can make them smarter. You can really make a difference. I think you can really make a difference. And individually, we can do that. We've got power. And after all, there really is an artist in science in all of us. And you don't have to be taking an art class with credentials and lots of experience. You don't even know how to draw. Heck, you've already seen you can have our voice in a lot of different ways. And that's why I say it's the art and science of you. It's your DNA. It's really the science that's in you. We're all genome explorers, and I hope by now you feel like we know that we're just on the edge of this. We're all really in the avant-garde. And when your story, put together with your unique voice, really has a power to be a champion for science, and you can really show not only the value of scientific research, but the beauty, the real beauty of it as well. So now just a few thank yous. Dr. Pai, who is really the PI and the leader of this group here at Fellman. And also I worked with Dr. Poani. Yeah, Anna, I know. But she and I worked closely together to refine a lot of the work that I did. And Dr. McGinnis is here, Jean. It was great to work with him too. And Denise was behind the scenes making a lot of things happen with details and such. And Jen, I'm sorry, Dr. Kovacs, I didn't get you on the screen, but she's a key part of this. All right, but still, you're important. And like Dr. Pai said, Nina Jablonski was part of the original research. And that's what I'm showing you here, this group. Nina is in the purple. And this was the core group at Nescent way back before we got to the curricular development.
5 years ago.
5 years ago. Thank you for your time. That's it. That's my presentation. I'll take questions. Okay, you got to ask me questions. There we go. All right, shout it out.
This isn't really a question, but I'm from Minneapolis, Minnesota, so I was really familiar with your work. So I have don't live too far from Iraq, and so I was like really familiar with that. So,
How about that? Yay, Minneapolis. Okay. Yeah. And so when you see Congressman Keith Ellison on TV at CNN, ever, you've seen him, he's head of part of the DNC. He's from North Minneapolis, that's his area. But how did I do? Did I tell it pretty straight, pretty much how you knew it?
yeah, because when I had a couple of programs at Iraq, I'm from Golden Valley, so yeah.
Okay. Great, thanks for saying that. Yeah?
Okay, so I'm not a bio major or anything, so I came out of curiosity, but I can't link to, so sorry if you answered this. So you say it's like everything, the origin of Africa, correct? Do you know where specifically? Because I heard the baby's like Egypt or like, in terms of like where.
That is one of the key questions, and maybe in your lifetime we will really be able to figure that out. We've got fossils all over Africa. They have all different morphology. Can I go ahead with this and correct me when I'm wrong, really? Because you know that my background is studio arts, and I've read and learned and been a student. This is what I do know. All over Africa, they're finding, and these are rare fossils. These are like treasures that they find, and they find them with tools. Sometimes the more recent hominids. And if you've been following what's happening with Homo naledi in the star, what's it called, the system with Berger. Anyways, very strange morphology there. Very a mixture of very ancient with very modern. Kinds of things. Okay, so we've got all of that information. Then now as we're putting together with the genetics, the thing is, we don't have a lot of genome sequenced from people living today in Africa, and we're just starting really now, really at the baby steps of ancient DNA sequencing of a lot of population groups. So this is an exciting time, and a lot of you maybe, hopefully will be adding research to this, but there's papers coming out all of the time with this. To answer your question, the origin of humans, it's Africa. That's our homeland. We know that. But exactly where in Africa? South Africa? Way down at the tip? Up close? Where we know that the launching to get out of there into the rest of the world had to go through the Saharan Gateway, into Israel, and then maybe around the Beachcomber route all the way to Australia.
And also,
Yeah.
just like,
Okay.
random question, I don't think you'll know the answer, but they found like a fossil that was like this big. Did you hear about that? It's like a skeleton, and it was like the person was like this big, and they're like, it's an alien. I don't. No, I don't know. It's real. Like you can look like they found like a little like.
Well, you talk about the Hobbit. The Hobbit's the little Flores, the island of Flores in Indonesia. Okay, so we're thinking, and there's been some recent interesting papers on this because they speculated on when these folks lived, and they thought they were maybe quite ancient, but the recent papers show them to that they lived until recent times, but it shows that they died off much further past in time. Okay, so I'm a little fuzzy there, but I'm trying. But also, there was this controversy supposedly between, well, did they have sort of a defect, sort of this characteristic with their brains that made them just diminutive? Something wrong with them as a disease kind of thing that made them small? Or was it the island effect? So you all know what that is, right? Population gets on an island, they become isolated over time. They sort of switch it up. Little creatures become bigger, and the big ones get smaller, trying to adapt to the environment and limited resources. They're thinking it's the island effect. The problem is with that, and it's a gorgeous cave. If you look on the internet, you can look for the scale, do the Hobbit genomics, whatever. Is that it's wet there, so they're not getting really DNA data from this.
They're a little bit bigger. They're not quite.
Yeah, good. Thanks, Jen. Yeah. Well, you're shorter than me, so whoa. I'd like to meet them. Okay, there's got to be more questions. Did I mix up on any of the science? Because you all are knowing things.
Okay, I see a hand. Yeah.
So, which
would you say that there is a service that is better than the others for genomic testing, like 23andMe versus Ancestry versus any of the other ones? Would you say one's better than the other?
Well, I do have opinions, but I can point you to Dr. Charmaine Royal's research. She's at Duke University, and her area of research is all about connecting genomics with race, but looking really skeptically at genetic testing. So I've got a couple of links to her research that if you want, send me an email, I'll send that to you. But then what I found is that these kits will have sort of a speciality. So Genographic Project is that deep ancestry story. It's haplotype information that they give you, and they really show that human migration. So it's really molecular anthropology, changes over time, which I think is actually most beautiful. But if you want to look more about who you are, say with an African American questions, then Ancestry.com, Ancestry has been, I think that the gene sets that they look at there have more of a population from those groups that would have more of the information. 23andMe has really expanded. I think they do a pretty good job with Ancestry, but of course now they've got some medical stuff too. And don't you love the way 23andMe shows the information, the graphics, with all the autosomes and how you can pull out some of the sort of the regional areas? Yeah? Okay, I hope I've inspired you to mix this up, talk to people, be advocates for science, be champions, go out there, make a difference. Thanks.
Thank you.